Why is detailed tissue analysis so crucial in histology and pathology? To unravel the mysteries held within cells and tissues, scientists and medical professionals rely on sophisticated tools and techniques. One such instrument is the freezing microtome, a device that has revolutionized the way we examine the microscopic universe within us. The freezing microtome is a specialized piece of equipment designed to slice ultra-thin sections of frozen tissue samples for microscopic examination. But how does this process work, you may wonder? The journey begins with a tissue sample, which is carefully embedded in a medium that freezes rapidly. This swift freezing process is key, as it preserves the cellular structure of the sample, allowing for a more accurate analysis. The tissue, now frozen and ready, is then placed onto the microtome. The microtome, a device with a precision blade, then gets to work. It meticulously slices through the frozen tissue, creating thin sections that are ideal for microscopic viewing. Each slice is a delicate window into the inner workings of the cells, a snapshot of their state at the moment of freezing. These thin sections are then carefully collected and prepared for microscopic examination. Each slice is placed on a slide, stained to enhance the contrast and detail, and then covered with a cover slip. The slide is now ready to be viewed under the microscope, revealing the intricate details of the tissue sample. But why is the freezing microtome so important in the fields of histology and pathology? The answer lies in the unique benefits it offers. The freezing microtome is particularly useful for samples that are sensitive to the chemical fixation process used in traditional paraffin embedding. It allows for the examination of fresh tissue samples, or samples that need to be analyzed quickly, without the time-consuming process of chemical fixation. Moreover, the rapid freezing process preserves the cellular structure, providing a more accurate representation of the tissue. This can be crucial in diagnosing and understanding a range of diseases, from cancer to neurological disorders. The freezing microtome also allows for the preservation and study of enzymes and other biochemical substances that may be lost or altered in traditional preparation methods. In summary, the freezing microtome is a vital tool in the arsenal of histologists and pathologists. It offers a way to delve deeper into the microscopic world, providing a clearer, more detailed view of tissues. By aiding in the diagnosis and understanding of diseases, it plays a crucial role in the advancement of medical science. So the next time you ponder the intricacies of the human body, remember the freezing microtome. This humble instrument, with its precision blade and freezing capabilities, is making waves in the world of science, one thin slice at a time.